Hello, everybody. My name is Stephanie Strathy. I'm the Harold Simon Professor at UC San Diego in the Department of Medicine and co-director of the Center for Innovative Phage Applications and Therapeutics with my colleague, Dr. Chip Schooley, who's one of the panelists today. I'm just delighted to have all of you join us for what is going to be a very special event, our Salt in My Soul panel discussion. Thank you for all for making time for this important conversation. As you know, I played a small role in this story today. My husband actually was the first patient at our center, IPATH. He was dying from a superbug infection in 2016 and was successfully treated with phage therapy by Dr. Schooley and a global village of phage researchers. This is how Mark Smith, Mallory's dad, heard about phage therapy from my husband's story that went viral. And he reached out to me and asked for help when Mallory needed it. So the story began. I'm just am really deeply moved to have the privilege of being part of this event today. And I want to thank the Smiths, Diane and Mark, for making their inaugural pledge to IPATH because they have made all the proceeds from Mallory's book, Salt in My Soul, and the documentary. All of those proceeds are being dedicated to phage therapy. So today I want to thank the Harold McAllister Foundation and family for their incredible ongoing funding of IPATH, as well as David A. and Karen Richard Sachs for making film copies available to all of the participants today and for supporting phage therapy research. We have an amazing group of panelists. We're going to have a Q&A after they introduce themselves. I want to remind everybody that we are recording this event. It will allow you to view it later on our website following the event if you missed any of it. And also you can type any questions that you have into the Q&A feature and we'll address as many of these as possible during the event. Now to start our program, I'm going to ask Diane Shader-Smith, everybody's wing mom, mother of Mallory Smith, and Will Battersby, director of Salt in My Soul, to introduce themselves.
I'm Diane Shader-Smith. I'm Mallory's mom, and I wanted just to give you a little bit of background. Okay, So before Mallory had her memoir published posthumously by Random House as Salt in My Soul, I was approached by filmmakers who wanted to tell her story. My initial reaction was no, which really meant not yet, because Mallory was a writer and her story needed to be told first in her words. She was convinced that patient voices needed to be amplified and had spent 10 years secretly documenting her treatments and hospital stays, the love and unexpected community she found in hospitals, the transcendent intimacy she developed with her beloved doctors, and the problems imposed by the medical system she depended on. Mallory was fascinated with the intersection of human behavior and the environment, and she wrote about human sickness and environmental destruction. But she was also just a girl writing in her journal about typical coming-of-age issues, the importance of deep and meaningful friendships, her longing for love, and most heartbreaking to me, her intense desire to have a child. After a year on the road sharing Mallory's story, it was time to reach audiences that might never read her words. It was time to say yes, to let the world meet Mallory, the young woman who shows us the power of personal story, the patient who teaches healthcare professionals that if you're really going to deliver patient-centered care, you have to listen to the patient, the environmentalist who wanted to heal the planet in some small way through her words and her work, the girl who prioritized love above all else, but the daughter that I lost too soon. So Will, he came into California and did a round of interviews. When it was time for him to fly home, I told him that he would be back, that there was so much more to the story than we had ever talked about, and that we could have possibly talked about in our first meetings. And so I promised him that he would feel the need to come back. And besides, there were additional people to interview, like Stephanie, for example. But then COVID arrived on the scene, and that became impossible. Trust was always an issue for me in the beginning. How could this man who didn't know Mallory, who had just met us, who wasn't part of the CF community, possibly tell her story? I took a leap of faith and I did a lot of prodding, which led to endless hours on the phone, talking about what the film was going to be about: visible and invisible illness, Mallory's writing, human health, environmental erosion, mental health, antimicrobial resistance, phage therapy. Will pushed me hard to comb through my files, photos, and videos. He told me to pressure my friends, and all of that led to a wealth of previously unseen material. With so much archival material to work with, which way was he going to go? At some point, I started to let go of the feeling that I had to control things. Will had earned my trust. And then he delivered his beautiful film. We all held our breath until the reviewers one by one filed their stories. Rotten Tomatoes has given it 100% fresh, and critics have called it a powerful film, emotionally affecting and profound, unflinching, and genuinely poignant. Will has done more than given us a beautiful film. He's given me a platform, a way to reach people, to use the film to change the trajectory for patients with CF and for those fighting a superbug. As Stephanie mentioned, we're donating profits, raising money, and raising awareness. Tonight is an example. We are so honored that IPATH is using the film to bring everyone here together to talk about phage therapy, what has become one of my favorite subjects. Thank you.
Hi, I'm Will Battersby. I'm the director and producer of Salt in My Soul. I was first introduced to this book, this extraordinary book, at the end of 2019 by one of the other producers, Richard Abate, who had helped Diane sell Mallory's posthumously published memoir, Salt in My Soul, which was taken from a 2,500-page secret journal that Mallory had kept throughout her struggle, and in which she documented this, this extraordinary life. And I knew right away that this would make an amazing film. And as soon as I started speaking to the Smith family, and I got a sense of Diane, one of whose nicknames is the Mamorazzi, how much she'd actually documented, I started to suspect that there would be an amazing movie in this. I then, we dove in, as Diane said. I prodded, pushed, demanded more archival, and what slowly emerged was that Mallory had not only kept this extraordinary memoir, but she had also recorded hours of audio. She'd recorded video, secret video of Diane, one of which is Diane dancing in a hospital when she didn't know Mallory was filming, which she wouldn't let me keep in the final film. But so, and thus began the journey. I knew what we had was, a very cinematic story, and so I really set out to try and avoid, the pitfalls of making a very didactic or expository documentary, and really stick to, the story of this very simple life. And then that's how the journey began, and more to come. But back to you, Stephanie.
Thanks very much, Will and Diane. I'd now like to ask each of the panelists to introduce themselves, starting with my colleague, Dr. Chip Schooley.
Hello, everyone. I really appreciate all of you being with us again today, and I want to extend my welcome to each of you. I also wanted to let you know this has been an incredibly moving story from the beginning, and I would encourage those of you who haven't had an opportunity to read Salt in My Soul to do so, and to see Will's incredibly powerful movie that took this from words to the screen. I just finished watching the movie today, and it was one of the most solemn experiences of my life that really helped me understand the depths of Mallory's feelings about life, and learned a lot about her, but also about me and about all the patients that I've taken care of in my career. It's something that everyone in healthcare, or who has a friend or family member who's ill, should really consider seeing. I got involved in the story as an infectious disease physician many years ago, when I was trying to decide what to do with my life. I was a resident trying to decide what specialty in medicine to go to, and I liked infectious diseases because people who had bacterial infections could be treated. We had drugs that worked for infections. Many of my colleagues who were cardiologists and neurologists had people who had terrible diseases, but didn't have medicines that worked. It was dramatic when someone came with an infection. You picked the right antibiotic and they went home. Over the course of my career, this became eroded over time as more and more multi-drug resistant bacterial infections emerged with our overuse of antibiotics in hospitals, in animal feeds, in our environment in general. And by the time Stephanie and Tom Patterson came home from their trip to the Middle East, it was not infrequent that we would find bugs in the hospital, so-called superbugs, that were extremely difficult to treat or impossible to treat. And it was out of this that the interest in phage therapeutics has evolved. This was a forgotten field for many years, but the ability to apply scientific principles to it has finally arrived with with advances in technology, and the Tom Patterson's case and Mallory Smith's case taught us a lot and reignited a field that I think will have a lot to offer people with multi-drug infections over the course of the coming years. So let me stop and turn the microphone over to my colleague, Doug Conrad. Doug?
Oh, good afternoon. I'm thankful for the organizers, for IPATH, for inviting me onto this panel. I totally appreciate it, and for bringing me into contact with Mallory, who I've heard about but never had a chance to really get to know. And I feel like the movie does a phenomenal job of telling her story. And to let you know a little bit about me, I'm a professor of medicine. I'm a pulmonologist. I started working in CF in the fall of 1989 up at UC San Francisco, and I came down to UC San Diego in 1996, where I started the adult CF program and have been working as in CF as a primary focus of my professional career. I have supervised close to 3,000 or 4,000 of these pulmonary exacerbations over my career. I first got involved in phage biology with a collaboration with a remarkable group of phage biologists, also involved with IPATH at San Diego State University. At the time, we were interested and still are interested in the community dynamics, the community structure of the CF microbial community, and more recently, the metabolism of it. This group had been focusing on coral reefs and the phage interaction and the health of our coral reefs around the world. And I think it's a story, my involvement in this group would have thrilled Mallory because my lab meetings frequently involved conversations about coral reefs and CF at the same time. So anyways, that's kind of where I am right now, and I'm glad to be here.
Saima, it's your turn to introduce yourself. Great, thank you.
Yes, so thank you. So my name's Saima Aslam. I'm a transplant infectious diseases physician, and I've been working closely with phage therapy since 2017. So it's been an ongoing experience. I really found Mallory's experience, both the written book as well as the recent movie, something that I think many of my patients share as well. I work closely with patients that are pre-transplant as well as after transplant. And so, you know, a number of them have had long hospital stays and experiences similar to Mallory's. And I think, you know, reading the book and again, watching the movie kind of brings home to me what my patients experience. And it sort of makes me feel very privileged and fortunate that I'm currently at a stage in my career and at a place where I'm able to offer phage therapy to so many patients, usually in the setting of transplant, including, you know, actually recently a young woman who had CF, you know, and we sort of treated her with phage, which allowed her to undergo a successful lung transplant, you know, which I think is something Diane also would appreciate. And hopefully it's something we're trying to make more available for our patients, as well as ideally at some point have so much more rigorous data. So it's available not just for patients in San Diego or at select centers, but really for people wherever they are when they need it. Thank you.
Thanks, Dr. Aslam. And David Pride.
Hi, thank you so much. It's really a pleasure to be here with you all. And while I don't have any very specific interactions or didn't have any specific interactions with Mallory, I have to say, it's these sorts of stories that really inspire us all, myself and Sima and Chip and Doug and Stephanie, to work really that much harder at the things that we do. So I am an infectious disease doctor here at UC San Diego. I also am heavily involved in running our hospital's clinical microbiology facility. And that's something that sort of puts me in a bit of a unique position, and it puts IPATH in a bit of a unique position because we have access to, all of these pathogens that are making people sick, whether people have cystic fibrosis, whether they come into the hospital for one reason or another, we've got access to these isolates. And that's just incredibly valuable for us in terms of being able to find bacteriophages that kill these pathogens. So I work largely in a capacity with Chip and Stephanie in directing the IPATH laboratory. And one of the main things that we're trying to do here at UCSD is to build out a bacteriophage library so that we can kill many of these pathogens that are making people sick. We've had some successes, some things are harder than others, but we really, in a myriad of different ways, are trying our best to sort of build out this so that there are fewer and fewer stories of people, for example, who have antibiotic resistant pathogens that we just don't have answers for. So I'll turn it back over to Stephanie.
Thanks very much, Dr. Pride. So at this point, we're going to have some questions to the panelists that we have prepared and that we've received from audience members previously. If you have any questions that you'd like to ask any of the panelists, either in general or specifically, you can enter these, just type it into the Q&A and we'll try to get to them. If we don't get to all of them, we'll follow up with you afterwards and make sure that we get an answer to your question. So my first question is for Dr. Conrad. Can you say how you think that Mallory's case might be able to help or inspire other CF patients?
Yeah, absolutely. So for the listeners who haven't read the book or seen the movie, I strongly encourage you to do it. It's a remarkable story that just lets out all of her, exposes all of her concerns, her fears, and also her hopes and her vulnerability. It's really quite of an amazing story. And I would say that despite all the recent advances that we've had, and we've had some remarkable therapeutic successes in the last few years, the clinical course that Mallory went through is still occurring. And it's not just the patient, it's other really highly resistant bacteria, including Pseudomonas or Chromobacter and Stenotrophomonas multifida, for instance. I think why this story is so critically important has everything to do with Mallory and her eloquence, her ability to express herself and connect with her listeners and her readers. And that is a gift that I have seen in very few, if any, of my own patients over the last 20 or 30 years. And I think the movie and her story in particular just highlights how important advocacy for our patients are, okay? And this comes through really strong in the movie by her parents, her family, and her friends, and how they were willing to pursue all avenues, even when there were like what seemed to be insurmountable barriers. I think a good example of that was their search for a transplant. I have been through that very same scenario with other patients trying to find the right transplant center, and the whole odyssey that led them to eventually think about and try phage therapy. I think these kind of examples, these stories are all very inspirational for a lot of the patients who are seeing her story.
Great. Thank you very much, Dr. Conrad. And now for Diane. How are you using not just Mallory's book, but now the film to make a difference and to advocate for phage therapy? You need to put your mic on. Sorry.
Thank
Thank
you.
you.
Thank you. I've been giving book talks for 3 years, and I've done over 200. And every time I spoke to a group, 90% of the time they had never heard about phage therapy. So specifically, in addition to all the other messages, the book became a platform to introduce phage therapy. And what I learned from Will's trailer, he produced the 2-minute trailer, and all of a sudden, in an instant, like literally like within 48 hours, that trailer went all over the world. And we started getting requests from Australia and Poland. And what I realized is the power of storytelling, using a single story, a single voice, a single case to underscore all of these larger issues. And I think that the reason that her story is resonating is because people see themselves in her story, whether it's fighting with the insurance, which is in the book, or whether it's in the film when she's expressing how she feels about having to be dependent on her parents. I mean, all of the different issues that she goes through, I think, touch different people at different points for different reasons. And I think it's the sort of combination of everything that has enabled people to find something in there that makes them want to work towards. But the phage therapy piece was sort of an interesting surprise because it was Mark's journey. My husband, dug deep looking. He was looking into pig's lungs, and he called George Church at Harvard. And he's the one who said, talk to John Dennis in Alberta, Canada. And it went on until he connected with you from doing his research, Stephanie. And then Stephanie sent out the tweet, and that tweet, it's the tweet that changed the world. Stephanie suggested we reach out to somebody at Twitter, and we did. We actually got a response, and they said that a lot of the medical professionals now are using Twitter as a forum. But I do think that these types of forums that bring people together, I mean, we could have done this at any point, but it's Will's film that brought us all together. So I do think that the medium of film using personal story is a really effective way to spread the message.
Absolutely. I couldn't agree more. The next question is for Dr. Schooley. You are going to be leading the first NIH-funded clinical trial of phage therapy in collaboration with the Antimicrobial Resistance Leadership Group. Can you talk a little bit about that trial, when it's going to start, and why do we need clinical trials in order to move the field forward?
One of the most important things for physicians when new therapies come about is to know how to use them best for the largest number of patients. And we can have stories like Tom Patterson, who had a great result, or Mallory, who unfortunately didn't. And we can learn a lot from that. But when we have systematically performed trials, we can develop information that helps us generalize how best to treat much, much larger numbers of people in the best way. And that's why these clinical trials are important as new therapies come along. And that's why the FDA uses them to decide about approving the use of drugs in the United States. In the case of this trial, almost all of the use of phages in the past has been in the presence of antibiotics. Antibiotics also kill bacteria, and it's hard to separate out the activity of the antibiotic and the phage to understand the independent activity of phages so that we can use them with more intelligence. And this is a study of healthy people with cystic fibrosis who are volunteering to take a single dose of phage and let us measure the impact of that phage on the bacteria in their pulmonary secretions, so we can understand in a very quantitative and precise way how best to plan to use phages in many, many other clinical trials. The study is one that has been enabled by support from the federal government, which is important because that draws in interest from other entities that invest in new therapeutic development, like drug companies, which is very important here. It has been important to have it be done by the Antibiotic Resistance Leadership Group, because this is the group of people in the US who lead the most advanced studies of all of our new antibiotics. So this is introducing phage therapeutics to the group of people who are going to take them forward across the country and across the world, because this is what they've been doing for their entire career. We anticipate the study will start in the next couple of months. We've been working very hard to have everything, all the T's crossed and the I's dotted. It's a multi-center trial, which will start at multiple centers simultaneously, so we expect it to finish very quickly. Dr. Pride is going to be leading the laboratory evaluation of this with support from Diane and her foundation, which is really a big help to us in the trial. And we think this will really provide much more insight into all of our other uses of phages and be a new step on the road to much broader availability and approval by the FDA.
Terrific. That's a wonderful advance. I know that the NIH has spent at least $20 million investing in phage therapy, and it's largely to the success of people like you and Diane who've been advocating for more research in this area. The next question is for Dr. Pride. Can you explain to the audience members why people need to go on a phage hunt? What is entailed in that?
Sure. So going on a phage hunt is basically trying to find a specific phage that is capable of killing a specific pathogen. Put simpler, if there's a bacteria out there that's making someone sick, let's find a virus that is capable of killing that bacteria. And while there are a number of different bacteriophages out there that are capable of killing these bacteria that make us sick, I think the average person would probably be surprised that we really haven't taken much advantage of it. So for example, you can look in any source of water, you can look in the dirt, etc. And these bacteriophages are literally everywhere, but we just haven't really thought, hey, can we use these in a way that may actually be beneficial for people? So what we've been doing in IPATH is we've been collecting these pathogens that make people sick. For example, for the Pseudomonas clinical trial that's coming up, or for the cystic fibrosis clinical trial that's coming up, we've been finding these Pseudomonas isolates from our patient population, both those who have cystic fibrosis and those who do not. And we've been just going out to, riverbeds and sewage sources, et cetera, and finding a bunch of different viruses in those sources that are capable of killing these bacteria that are making sick. And some of them we find are actually really good at it as well. So that's one of the biggest points that we would like to make, is that sometimes these things can be relatively easy to find. Once we find them, of course, we have to characterize them, make sure they're safe, et cetera. So in some ways, that's kind of when the hard work actually starts. But the nice starting point is just that we're going out and we're finding these bacteriophages in very common sources in our phage hunts and find that they're capable of killing a lot of these bacteria that are making us sick.
Great, thank you very much. The next question is for Dr. Aslam. Dr. Aslam, you're working on specifically on building a registry and a library specifically for Burkholderia cepacia, which is the organism that really took Mallory down and really affects a lot of other CF patients. Can you talk about the importance of that kind of a registry and how you're leveraging so many researchers from around the world?
Yes, thank you. So with a generous gift from the Mallory Smith Legacy Fund, we've set up a registry. It started sort of right in the middle of COVID, so it took us a little while to get going. But the goal of the registry is to have sort of one site where we can collect information of CF and lung transplant patients that are infected with Burkholderia. So Burkholderia is a highly drug-resistant organism. It's very rare. It's only about 3% of CF patients have it. And because it's rare, a number of drug companies are sort of not going after it. So for that, that's not a priority for them. And I think that's why, in particular, the Legacy Fund is helpful to help us get going. But our goal is to get information on patients that have this infection, to get their bacterial isolates. And we're working with a research group in Israel, Dr. Ronan, and with his lab. So we can sort of collect all the bacteria that are drug resistant from these patients, funnel them to this one lab. There, they're actually working and looking for viruses or bacteriophages that can kill the Burkholderia. So currently, our goal is to develop, a wide library or bank of these Burkholderia phages. So when a patient like Mallory pops up and they need a phage, we actually have a place to send that bacteria and already have a group of like 60 phages that we can run against and find something that will treat that patient. Instead of a Twitter hunt, which was wonderful, but it sort of went all over the world and kind of back again. But here we're trying to put all that information, all those phages in one area so that when a need comes up, we're actually able to go to that one source. And so far, we actually currently actually have a patient in Montreal we're trying to help that we found phage for from that library that we're planning now to actually manufacture and treat the patient with.
That's terrific. Thank you very much. And I guess this is a question for any of the panelists. In addition to specific clinical trials like the one that Dr. Schooley and Dr. Pride are involved in, what other initiatives can people undertake to try to move phage therapy forward, given that we're facing a situation where we have over 1 million people dying per year from superbug infections, and by the year 2050, that's going to be 1 person every 3 seconds. Who would like to take that on? That came from one of our audience members.
I think there are many ways that people can help with this. One is advocacy, because I think the people underestimate the impact of multi-drug resistant infections, and they occur for bacterial infections, mycobacterial infections, viruses. Anything that you bring an antimicrobial to the table for will develop resistance over time. And so people need to understand that ongoing drug discovery, drug development is going to have to occur for us to stay ahead of the evolution that's been going on for hundreds of millions of years among the pathogens that cause us illness. This may not be as topical or sexy as a new outbreak like coronavirus outbreak or Ebola, but it's happening all the time, all the way around the world, and it never stops, as opposed to some of these outbreaks that come and go. So it's really critical for people to be able to discuss broadly to our politicians and those who allocate resources the urgency about investing in this. There are other places where drugs, antibiotics are used that really we should think about. Antibiotics are used to help chickens grow more rapidly. That ends up helping a few flocks of chickens get to the market sooner, but it also puts antibiotics that aren't degraded into the environment that then induce resistance, both in those chickens and in the wastewater around the chickens that come back to us. And so there are a lot of things that I think the public, as they learn more about it, can help us spread the word about. Let me turn the microphone over to others, because other people have been thinking about this too.
I would add to what Chip said and take maybe a slightly different angle on it. And that's that one of the things that sort of slows down the development of phage therapy is just the idea that phages aren't that available. And it's a little counterintuitive to me, at least, because phages are literally everywhere, but we don't have a well-characterized bank of phages capable of killing all these bacteria that make us sick. So at IPATH, that's really one of the things that we have kind of dedicated ourselves to doing, is to creating and characterizing that type of a bank that will make phage therapy at least a little bit easier for people as they move forward. And the characterization can be very, very difficult for some of these phages, knowing whether they're safe to put into people, et cetera. So all of that hard work really has to be done on the back end, really, I think, before people, particularly including the general public, can feel comfortable that this is something that can be both safe and effective. The one thing that I would advocate for, though, is the idea that for a lot of these pathogens, phages actually aren't that difficult to find. They might be a little bit more difficult to characterize, but they're not that difficult to find. So one of the ways that people can get involved, particularly if they're in the right age range, is just to volunteer and help find phages. There are all sorts of programs like C-Phages programs at IPATH. I mean, we certainly take volunteers who have just been excellent at helping us find additional phages that we add to our banks and we characterize and keep this whole thing moving forward. Maybe slower than we would like, but it's still moving forward.
Thanks very much, David. And
Stephanie,
yes, go ahead.
I wanted to weigh in on that because something very interesting happened to me. I raised a lot of money in Mallory's lifetime, and when she passed away, a lot of my friends who were the bigger donors said, we donated to Mallory's Garden because of Mallory. They didn't really have any connection to CF. They said, please don't hit us up anymore, which is why I stopped Mallory's Garden, switched to Mallory's Legacy Fund to just really raise money for phage therapy. And when the Lancet study came out and when I sent it around and posted it on Facebook and I started quoting the statistics that you were rattling off and making people understand that it is not just for CF. And in fact, CF is a teeny weeny little piece of it. That really helped because I have had a bunch of donations and every day now, since the movie's come out, I get an email that says somebody's made a donation and 9 times out of 10, I don't know who they are. And I think it's because if we can make the case that phage therapy will help all of the regular people, the non-CF patients who will be encountering a superbug. I got a $25,000 donation from somebody whose husband, who is perfectly healthy and they're very wealthy, recently had a lung infection that became resistant and he had multiple rounds of IVs and it wasn't working. And I called her during that time and I made the case and she gave us 25,000. So I think taking it out of the CF population, even though that is where my heart lies and where we ultimately want to be and where we're going to get the patients from for the trials. And through CF Mamas, I'm on there every day mouthing off about PT and raising money and pound your doctors and tell your centers and ask people to give. So I think it's got to be a combined effort inside and outside.
Thank you. You really are our wing mom, Diane. There's a question from Will, from one of our audience members, that, this is obviously a film that is serving many purposes. It's a personal story. It's an honoring of Mallory's memory. It's about advocacy, and it's also about science communication. How do you balance all of that? And what was the aha moment for you?
I wish I could say I'd done it all intentionally, but that would be a lie. I set out to make the best film possible from this extraordinary life, and I think, it's true of every chronically ill patient, right? That goes through these kind of things. There are amazing ups and downs, right? There's and I think what was special about Mallory, which was said earlier, was really how she chronicled it. But I think that all patients live these kind of lives. So if you're going to, you've just got to tell those stories. And I think the implications of those stories become very apparent and very powerful. And I think what's been, the big aha moment for me is really, I made as kind of, beautiful and poetic a film as I know how. But what's been amazing is how many groups have said, oh, I see, this is a film about antimicrobial resistance, right? Environmental groups have said, oh, this is a movie about environmental degradation and our reciprocity with nature. Mental health groups have said, oh, this is a movie about mental health. But of course, all patients are facing all of those issues. But I think that's been, so yeah, I don't know that I'd call it an aha moment so much as I'm very proud of that, right? That the film seems to be having, it's giving people a platform and a springboard to talk about their issues. And I think that's the sort of the power of storytelling, right? Within medicine, within academia, right? And I think, the more you move people, the more you make people feel about a subject, the more you're going to impact them and draw them in. And, and I can't tell you how many conversations I've had about phage therapy, which I had never heard of 2.5 years ago, and that's been wonderful.
Super. Thank you very much. Dr. Conrad, we had a question from the audience. If you could talk about what kinds of infections that CF patients have in particular that phage therapy might work for. So we've heard about Pseudomonas aeruginosa, we've heard about Burkholderia cepacia. Are there other kinds of organisms that may affect CF patients that phage therapy could assist?
Yeah, absolutely. There's and this story is actually changing over time. As many of you know, there's been a therapeutic breakthrough a couple of years ago with a drug called Trikafta that has really improved the outlook for many of our patients, and it is changing the microbial communities that we see across the country, but still. I would think the unmet needs remain Pseudomonas. In Southern California, we do not have that much cepacia. We've always been very low. All the centers, as far as I'm aware, it's mostly a problem in the Northeast and in the Midwest. We have problems with Achromobacter, Stenotrophomonas multifida. We have a big issue, as Chip mentioned, a lot of problems with mycobacteria. Standard E. coli are problematic also. So all of these are potential targets for phage therapy. I should also mention that there are just within ataxa, there are badly behaving Pseudomonas, and there are a lot of our older patients who are 70, 80 years old have had Pseudomonas for 30, 40 years. We've had Cepacia patients for 10 or 15 years. And then we put them on Trikafta, and then the Cepacia vanishes in less than a month. So there's a lot that we don't know about those microbial communities and how they're functioning. But
Great,
those are the
thanks.
major classes that we're looking at.
Yes, great. Thanks very much. And, it's also important to realize that there's lots of other kinds of patients that are affected by superbugs as well. We receive a lot of people who have urinary tract infections or prosthetic joint infections. Dr. Schooley, I know you have something to say too. Maybe you would also address some of the questions that have come in about the regulatory pathway and how the FDA is viewing this.
Let me just toss this to Dr. Aslam first, because I want to make the point that you just made. It's not just multi-drug resistant bacteria. It's also bacteria that get in places that are difficult to treat. When they get onto prosthetic joints, when they get onto pacemakers, they form biofilms. And the biofilms protect those organisms from killing by antibiotics and make it very difficult to sterilize these implants that many, many more people have these days than before. And in the past, we almost always had to remove them, treat the infection, and then hopefully be able to replace them. Do you want to talk about some of the applications along those lines that you've employed phages, Simon?
Yes, happy to do it. So we've actually treated many kinds of patients here at IPATH specifically, but also in collaboration with physicians at many other centers. So this includes prosthetic joint infection. So usually if a hip or a knee joint gets infected, the metal has to be removed and a new one put in. But we've treated at least one here and definitely more at Mayo Clinic in which the joint is just kind of washed out. We put phage in that as well as give it to patients intravenously to resolve the infection. I also, because I'm a transplant physician, we have patients that are on immunosuppression. So that at times makes it very difficult to resolve their infections. And also those patients tend to have really drug-resistant infections. So whether it's a urine infection or an infection of a transplanted kidney or a heart device in a patient that's undergoing heart transplant. So we've treated all of these kinds of patients with phage therapy, usually along with antibiotics. So the goal being to try and get synergistic activity. So sometimes there's synergy, sometimes they're not. But certainly we've had successes in cases where regular antibiotics have failed for years.
Great. And just to get back to the audience members that were asking about the FDA and regulatory pathway, we found that even beginning with my husband's case, that the FDA was actually very helpful. Sometimes they get a bad rap and that people have the impression that they're blocking progress. They've been very responsive. They haven't turned us down for a single request for phage for compassionate use. And they have also made it possible to approve entire phage libraries. One of our partners has a phage library that, and there is FDA approval to use the phage in that library in any combination. Where the regulatory pathway will be a little bit more difficult at first will be with genetically engineered phage and synthetic phage, because that's the future. Now that we have CRISPR-Cas gene editing to act as molecular scissors, we can optimize phage lifestyle, and that's going to be something that's going to be happening on the horizon. I wondered, Dr. Pride, if you want to comment on that. And also, if you may want to mention what kind of work can be done with respect to optimizing the synergy that we've seen sometimes between phage and antibiotics.
Sure. So, we've been doing a lot of work. There's other groups really around the country and the world who've been doing a lot of work trying to figure out basically how to take your average phage and make it better. One way to potentially make it better is to try to design something in a test tube that is better at killing the host. And that's sort of what we had referred to a little bit with the idea of synthetic phages. But there are other ideas involved as well, and that it may get to the concept that I think you guys heard one of us mention, and that is the concept of synergy, meaning that you could put two things together and get a response that is greater than each of them individually. So sometimes we look at the concept of, well, if one phage is good, maybe we can put multiple different phages together and we can get synergy, meaning that they're better together at eradicating that host than they may be individually. That concept also applies to the idea of, well, let's take a phage and let's take this particular antibiotic, and they may work together better than they might individually. And that is potentially something that we might have observed in some of Dr. Aslam's cases that she was talking about earlier, that maybe we are able to put a patient on an antibiotic plus give them a phage at the same time, and the sum of those two is greater than their individual parts, and thus we end up with people that could clear infections that we weren't able to clear using either just the phage alone or the antibiotic alone. So those are things that we're working on specifically in the IPATH lab, and we certainly hope that that they develop out as we build our phage library and characterize how things can work together.
That's terrific, David. I'm also aware that both the National Institute on Allergy and Infectious Diseases, as well as the CF Foundation, is funding research in this area, not just at IPATH, but around the US and internationally. And there's some very exciting initiatives that will really move the field forward in that respect. Is there anyone else that would like to comment on these issues?
I'll just say about the FDA. I mean, I think they have been really visionary in the way phages have come forward. They know how to balance risk and benefits. When you have patients who are as sick as Mallory was at the time the phages finally came together, they were actively helping us in terms of getting the regulatory documents in place so that they didn't hold things up. In the case of Tom, they not only helped us with the regulatory documents, they pointed us to another source of phages that we're able to use in this case at the US Navy. And they pointed me to their group that helps prepare materials for human use to talk about endotoxin and some of the agents that we needed to get removed from the phages. So they serve really not just as a regulator, but also as a consultant. But when you start doing clinical trials, they also understand that rigor is critical, because you want to make sure that when you're doing something elective and that when you're doing something that later you want to be able to generalize, you'll know exactly what you've done. And so they use the same kind of regulatory rigor with clinical trials that they do with phages, they do with other drugs and devices. And I think this whole spectrum of looking at things in this a very nuanced way is why they've been so effective in helping us move the field forward. So I have nothing but positive things to say about the FDA in the area of phage therapeutics.
That's terrific.
Completely
I'd agree.
Seconded. Sorry, go ahead.
Go ahead, Simon.
No, I was going to say, it's actually been amazing talking to the FDA for every single patient that we've applied, for permission to treat. And they've been amazing partners.
Yeah. I'd have to echo that also. I recently had an experience with a group phone call to get approval. It was 7:30 my time. That means 10:30 at night on a Friday night. And there was 3 people from the FDA giving me the last final instructions and wishing me well. So they've been very helpful in my experience.
That's terrific. We have a question from a phage researcher who I know from Twitter, who's asked about a specific phage D29 that she knows that is active against mycobacteria, but she's wondering if that example and maybe others like it are where you can see that a phage works in the lab in vitro, but doesn't necessarily work in the clinical picture. Do one of our physicians want to comment on a situation like that? Does that happen often?
I can comment, not specifically on D29, but phages in general when we treat patients. So I think in vitro, when we look at them, usually it's a very two-dimensional look in terms of bacteria and phage killing. But when we give it to a patient, there's the interaction from the patient themselves. So whether it's the patient's immune response to the phage or the immune response to the bacteria, which kind of helps modulate the response to therapy. So I think a two-dimensional picture alone isn't usually always sufficient to predict clinical success. And a number of times patients have multiple other issues, such as in a CF patient, it would be their lung function that feeds into how they're doing. Similarly, for other cardiac device infections, they also have heart failure. They have other things that may feed into how that patient does. And the infection is one piece of that puzzle and not the whole thing. So at IPATH, we get many requests, but part of our goal when we evaluate each request is to sort of assess the overall clinical picture. And if we think resolving an infection, would change the trajectory of the patient, but also how amenable we think that infection would be to phage therapy.
Terrific. We have a question from an audience member that's asking about what are the bottlenecks to moving phage therapy forward? We've talked about clinical trials. We've talked about the urgent need to develop a phage library. And here at IPATH, Dr. Schooley and I are working with some social entrepreneurs that are interested in creating a private-public partnership that would move that forward. But what other bottlenecks do we have? Dr. Pride, did you want to mention any that you're familiar with?
Well, sure. And some of it kind of gets to what Simon was just saying. And that's that sometimes it can be very difficult to predict when a phage gets into a person, whether or not it's going to behave like it does in a test tube. And one of the things that we would love to do is to study those phages against bacteria under multiple different conditions. Something just as simple as, for example, oxygen, which is readily available to us all. And when we grow a bacteria and we test to see does that phage kill that bacteria quite well, we usually do it in an oxygen-rich environment. And we don't necessarily think about the fact that when we inject that into a person, oxygen is not readily available. And sometimes those things really behave much, much different under different physiologic conditions. So one of the things that we're trying to do as we move forward our phage bank is to characterize the interactions between the host and the phages under a number of different conditions, obviously closer to the sort of conditions you might see in a certain part of the human body, to see, do these relationships fall apart under those different conditions, or are they quite robust, meaning that these phages may be really good candidates to put into people.
Great, thank you. Are there other bottlenecks, Dr. Schooley, that you would want to make people aware of that they might be able to help with if they had the resources?
Well, the original bottleneck with Tom's case was getting rid of endotoxin, which is made by the bacteria in which the phages are grown to be brought up to a large enough number to treat a patient with. There are a lot of ways to do that now. We feel pretty comfortable doing that. They can involve putting the phages in an ultracentrifuge and physically separating them. They can involve using an organic solvent that dissolves the endotoxin, leaves the phages back behind, but there are a lot of ways to do that. The area that we're most challenged these days is just what Dr. Pride was talking about, knowing exactly how to extrapolate, and Dr. Aslam as well, how to extrapolate what we've learned in the laboratory about a phage and a particular organism to the patient. The study we're planning on doing with the support of the NIH is going to look exactly at that question. We'll be able to look at how the phage interacted with that person's bacterium in the laboratory, and we'll be able to look and see what it actually did to the same person, same bacterium in that person. And by being able to connect the dots, we'll begin to understand which laboratory tests are most predictive of phage activity in people. It took us a long time to understand this with antibiotics, and I'm sure it'll take us some time with phages, but we'll use the same approach as we did, which is an iterative approach of going to the laboratory, seeing what we see, comparing that with what happens clinically, and then doing course corrections as the two areas begin to come together.
Great. I think it's really important that funds like Mallory's Legacy Fund are able to add to the CF trial that you're planning right now, to be able to support translational research, to be able to ask questions like, what kind of dose is the best for a patient with, this particular kind of condition, or what best route of administration should be offered. And this, what you've taught me, is that translational research goes from bench to bedside and then bedside back to bench. And that's really what we need to be asking is as you've said so eloquently in the past, we need to be treating phage like a living antibiotic and subjecting it to the same amount of rigor before we jump to phase 3 clinical trials, because the phage therapy field has had enough heartache in the past, and we really need to do it right next time. So I'm really glad to have you at the helm of this important trial, and to have the support of Mallory's Legacy Fund and also the CF Foundation providing ancillary types of studies that will inform the field.
I would like to say something. So one of the things that I do believe is that, and I've heard it because Will and I have done a number of talks and panels, and I would say at least 10 different researchers, whether it's Yale, Texas, England, all over, all of them say the same thing, which is they need money for compassionate use because none of the organizations will fund it. So I think the push needs to be made for that. And then also in general, they need more funding. And I know the Pasteur Act is in front of Congress, and Will and I are going to be doing a screening in DC with the pressure, to put the pressure on additional funding from the NIH. I mean, the fact that you have the data that tells you that superbugs are going to kill people, and the fact that the government isn't funding it doesn't really make sense to me. And I think it just seems to be that it has to be a priority for people in terms of the advocacy that they're doing and the fundraising and where the money is going. So I think fundraising is a bottleneck issue as well.
Absolutely. And that really points to the importance of advocacy, because legislation like the Pasteur Act, people need to know what's in it and what it means. And the initial versions of that only mentioned the new antibiotics. That language has been sufficiently changed now to talk about new antimicrobials that makes room for other alternatives like phage therapy. So those kinds of things are really important. People can get involved by talking to their legislators and making sure that they understand that this is very important to, improve and reduce antimicrobial resistance globally. I know that we're approaching the end of our hour right now. So if you've put a question in the chat and you didn't get it answered, we will be able to review it and get back to you. I want to take this opportunity to thank all of our panelists, and also the funders who have assisted us today. And I want to put a special thank you to Diane Shader-Smith, our wing mom. You've been a terrific friend and advocate, and we couldn't have done this without you. This was your idea. I also want to thank Lizzie Lampley. She's our IPATH coordinator. She starts work at 6 in the morning, and today we've kept her well overdue. And I want to thank all of you audience members for taking part. If you would like to assist us with our research at IPATH, there is a link. You can go to our IPATH website, ipath.ucsd.edu. If you have a superbug infection or know somebody that does, you can contact us at ipath@ucsd.edu, and that will reach simultaneously all of us. And we will be able to review your case. We are a nonprofit associated with UC San Diego, and we will do our best to help you. With that, I want to thank everybody. I think that today's panel has really shown you that even when you're faced with the most sorrowful tragedy, that it can turn out to be something that can save other lives. And truly, we have made a difference here. I really feel that Mallory's story and Tom's as well have really moved the needle, as one of my colleagues has mentioned it. And I think my tweet that went out trying to crowdsource funds or phage for Mallory years ago said, "This is what hope looks like." And that's what I'd like to end this with. This is what hope looks like, everybody. So keep on fighting, keep on advocating, and let's make a difference with superbugs. Thank you very much.